Welcome to KMINE web server instruction video. This web server can be found on kmine.tenwiseapps.nl and via our website www.tenwise.nl. In this video, I will show you our web server that allows you to search PubMed using our KMap database. KMap contains more than 200 million automatically mined literature relations. Your goal is finding new relations between your concept of interest with other concepts in just a few mouse clicks. You want to save your results for use in your in-house research pipeline. Now, let's explore the KMINE web server in detail. You can use the Info tab at any time to learn more about this KMINE server and the development and usage of the underlying KMap. For questions, feature requests, and other suggestions, please email us at kmine@tenwise.nl. On the start page, you can enter your biological concept here. Enter a keyword such as a human gene, bacterial gene, metabolite, pathway, phenotype, disease, drug, or organism. When you start typing, the keyword is automatically matched to the terms in our database. When you have selected the correct term, you can click the KMap Scoping button to jump to the tab where all relations to other terms in our database are shown. Or click the KMap Evidence button to show all abstracts sorted for evidence category. First, I will show you KMap Scoping by entering ulcerative colitis, like so, or UC. And if you then click on KMap Scoping, you come to this page with the tab Relations. On this tab, you see the relations between your concept of interest, ulcerative colitis displayed in color here, and over 200,000 vocabularies that are selected in the left-hand table. The total number of hits with your concept and the total of terms that are in the vocabulary are displayed in parentheses. In this case, there's a co-occurrence of ulcerative colitis with bacterial genes in 73 abstracts. And in total, we have over 7,000 bacterial genes in our KMap. The table in the middle shows these relations. It indicates per column name, name of the concept. Click on the header and it sorts alphabetically. Type, the vocabulary from which the concept is derived. Number abstracts, the number of abstracts in which both concepts co-occur. Score, a score denoting the uniqueness of the relation. The higher the score, the greater the uniqueness of the relation. I've selected now on score. The search fields can be used to search in the entire table for a keyword. For instance, we would like to know which microorganisms are related to UC. We do this by sorting here in the type table on organisms, and we score the table, and this gives a list. We are interested to learn more on the co-occurrence of Faecalibacterium prausnitzii with ulcerative colitis in PubMed. We select this row in the table and it directly shows the abstracts in the right results panel. In total, 44 entries. And by clicking on each arrow, you get the abstract immediately and you see depicted both concepts in color, ulcerative colitis and Faecalibacterium prausnitzii. Now, by clicking on the sentences button instead of the abstracts button, you will find the actual sentences in which both UC and Faecalibacterium prausnitzii co-occur in one sentence, giving you fast access to potentially interesting relationship by just looking at it. Abstracts that are found in this table can be selected by clicking on one or more rows in the table, like so. We select this one and this one and this one. And we add to My Abstracts by selecting the button, pushing the button, Add Abstracts to My Abstracts. The collected abstracts now can be visited anytime via the My Abstracts tab. Here you see them displayed. You can deselect them again from this selection. And using the radio buttons on the left-hand panel, you can format the output that you need. The default option is to show all the abstract with citation information, like so. But if you need an identifier, for instance, to import them into a reference manager program like EndNote, you can choose the PMIDS button. If you want to copy the reference directly into your document, you can select the citations option, like so. Finally, you can use the download button to download the abstracts to an Excel file. This file then contains all the information, including authors, journal, etc. Going back to the KMap Scoping tab, let us look at the other features. The second tab, Word Clouds. The word cloud will give you a visual representation of the data I've just shown you in the relations table. The font size for each concept is proportional to the score. And the two sliders on the right, can be used to select the maximum number of words per category, like so, and the scaling of the fonts, like so. Pictures of these word clouds can be copied and used for making presentations. The third tab I would like to show you is Network. This network shows the data from the tab Relations again in a network with the selected concept of interest in the center and up to a number of 50 other concepts per category, as displayed here. These concepts, dots, with their name inside, have a different color dependent on the type. In the right-hand above corner, a bar called Number of Connections per Category can be used to create and alter the network picture using between 0 and 50 concepts per category, like so. I would like to decrease this because this is a bit much. Decrease to 3 per category. The thickness of the arrows between the nodes indicate the relationship between the concept. The thicker the node, the higher the score on abstracts in which the two co-occur. By clicking on the arrow, like so, you are redirected to the TenWise server that depicts the abstracts with the co-occurrence. Again, highlighted in color UC IL23R. By clicking on the nodes, separate nodes, you are redirected to the same TenWise server that depicts the abstracts on this single concept. Finally, the network shape can be changed by dragging the nodes across the screen. Screenshots can be made and used for reporting. Going back to the Relations tab, there's another feature I would like to show you that enables you to, all by yourself, make a set of concepts that you are interested in. This is done by clicking the button Add Concepts to KMap Multiset. In our example, we make a concept set of our own around ulcerative colitis. I add that one. We already have been looking at Faecalibacterium prausnitzii, so I will select that one as well. It's over here, add concept to KMap multiset. Also, I would like to know more on the involvement of human genes. So we select the two human genes that have the highest score. In this case, NOD2 and IL23R. We are also interested in a known probiotic. So we select an organism, Rhamnosus, Lactobacillus rhamnosus, to the set. By now clicking on the KMap multi button over here, you see these concepts coming back. Now, click on X table or cross table, and you get the relationships between each individual concept that you have selected. Again, by choosing on each line, you get the abstract in which both co-occur, like so. And as before, you can choose to select the abstracts or the sentence mode, like so. The same table is made visual in the Network table tab. Here you see, for instance, that the co-occurrence between ulcerative colitis and rhamnosis is not particularly strong. However, both seem to have stronger connections to the same gene, NOD2, and the gut commensal Fecalibacterium prausnitzi, that seem to play an important role in UC. This could be a starting point for researchers to look if Lactobacillus rhamnosus could have an impact on ulcerative colitis via either of these routes. Finally, the same relations can be made visual in a heat map, as depicted here in this tab. Here you see the same relations and scores that display the relations. Now, I would like to go back to the start page and explore KMAP evidence. We have still selected ulcerative colitis, so we click on KMAP evidence and are redirected to this tab. This page shows abstracts for your selected concept, depicted here again in color, that is ranked between 0 and 1 for various types of evidence. This score is determined by running a previously trained random forest model on the abstracts. Evidence is defined as the biological context of the abstract in which the concept appears. For example, an abstract with a score of 1 for clinical is most likely about a clinical trial or an observational study with patients. Likewise, an abstract with a high score for mouse is most likely about a mouse experimental model. Types of model can be selected in the left-hand selection panel and are clinical, clinical patients, patient observations, variant, having to do with polymorphisms, DNA rearrangements, and mutations, microbiome, studies having to do with microbiota, metagenomics, and 16S profiling studies, mouse, experimental mouse models, studies on mice-derived data, see them coming, and finally, rat, experimental rat models, studies on rat-derived data. In all cases, the 200 abstracts with the highest score for the selected models are shown. Furthermore, you can narrow down or expand your search to a specific time period using the publication date slider. After adjusting the periods, a new selection is automatically generated based on score within this time period. To summarize, this generated table has the following columns: citation, the article in which your concept occurs selected for high scores with the selected model, score, the score between 0 and 1 from the random forest model, and year of publication, on which you can select. The search field button allows you to select words used in citations, and this can be any word including author names and journal abbreviations. Again, clicking the arrow before each row shows the corresponding abstract directly below. Clicking on the hyperlink, this number, brings you to the abstract on the PubMed site. Abstracts in the table can be selected and added to your set by pressing the button Add Abstracts to My Abstracts. You can revisit this tab as I've shown you before. This was the instruction video for KMINE web server. Please note that we are continuously updating and improving the features, and therefore the web server might be slightly different when using yourself. Want to get full access? Please contact us directly at kmine@tenwise.nl.